title of my talk today is Through the Unlabeled Lens of Spatial Multi-Omics. And so this was just a small part of what I'm going to be doing my postdoc in, and it's centered around the label-free imaging portion. So I hope you enjoy it. Oops, it's not. Oh, yeah, there we go. Really quick, are you able to see this laser pointer on the screen?
Yep, all good.
Okay, great. Really quick, this is my disclosure slide. I have to say it. And so the real motivation behind my PhD work is this quote attributed to Henry David Thoreau. It says, roughly, it doesn't matter what you look at, it's what you see. And in today's clinical landscape, just looking at a pathology slide and not seeing the underlying disease can have really huge consequences on the patient outcomes. And so to afford the highest diagnostic power, what is generally done is, at least in research and also some maybe advanced or rare diseases, is to take serial sections of a precious biopsy and apply a different set of stains to each one. And that's because our eyes aren't really the best at discerning between different shades of blues and purples and pinks that many of the histological stains that we use today are. But this comes with a lot of problems, including physical deformations from the slicing process, and it requires a lot of post-processing co-registration. And unfortunately, many of the structures of interest are limited to maybe one section and not present in other sections, meaning that you can't see that same structure stained with different, colors, or to observe different cell types. Additionally, immunofluorescence, even the highly multiplexed ones that was in last week's seminar by Julie, come at a cost as well, because a lot of the fluorophores are bulky, some of them are hydrophobic and alter the native distribution of the target. And as an example, like even some of the fluorescent dyes, promote, for example, lipid droplet fusion, precluding the use of these dyes in studying those really high resolution subcellular organelles. And so that begs the question, how can we detect both morphological and biomolecular information with high spatial resolution, but without expensive and sometimes toxic reagents or excessive tissue availability? And so today's talk is about this label-free platform, which is a multimodal platform composed of second harmonic generation, multi-photon fluorescence, and stimulated Raman scattering. So stimulated Raman scattering is a label-free molecular imaging platform in which it only requires the sample be placed in PBS or water just to keep it from burning. What we do is we overlap a pump and a Stokes beam in space and time, and then we use that to probe the row vibrational modes of the molecules in the analyte. And so that stimulated Raman gain or Raman loss gets collected by a photodiode, digitized into a signal interpretable by like a computer. And by tuning the wavelengths of these pumps and Stokes beams, we can selectively vibrate certain molecular bonds that we ascribe to certain biomolecules, such as the heme backbone or lipid tails or proteins in general. And what you get is what's called a hyperspectral image. So it's basically a series of images along the wave number spectrum, and each pixel has its own molecular trace. So what that enables you to do is that you can separate these, subcellular organelles because each, molecule spectrum will have a different amplitude and phase. And you can also directly, using unsupervised clustering, segment the subcellular organelles. And this becomes a really powerful tool when you don't really have the set of stains available to you to identify a certain cell type. You can, look at rare cells in situ or in this case in vitro to kind of figure out what kind of cell types there are, or even their metabolic state. And the other modality is multi-photon fluorescence. In this case, we use two-photon fluorescence to do optical redox ratio imaging. And that's because a lot of these labels like superoxide dismutase or glutathione labels are very expensive, and they're also very sensitive to other reagents that you might use typically. But the normalized optical redox ratio is kind of like a gift from nature, in which we just measure the flavin pools and the NADPH pools. FLIM, fluorescence lifetime imaging, takes us a step further. We can better resolve these pools, but this gives us at least an estimation of the oxidative stress within the sample. And the final modality, just for an ECM perspective, is the second harmonic generation. So fibrillar collagen, so like types 1 through 3 primarily, are really great because they have a non-center symmetric structure. So when you kind of irradiate it, or not, shine a light on it, you collect light with twice the energy. And this can be done in 2D or 3D. And so we can look at the collagen fiber angles, fiber density, fiber coherence, to identify, areas of fibrosis, and a lot of, biomechanical ECM changes happen in disease states. And so this is a really powerful tool as well. And so what we applied this to is this diabetic kidney disease question. So kidney disease is the 9th leading cause of death in the US. Has really, scary symptoms, but diabetes, for example, is a huge risk factor for chronic kidney disease, which a third of the patients develop, but it develops very slowly. And many symptoms like go unnoticed, for example, mesangial expansion, because there's not yet, at least in the US, a routine biopsy for kidney disease, and therefore it has a very poor diagnostic efficiency. So about 9 in 10 estimated with moderate to severe cases remain undiagnosed. So it's kind of a catch-22 because there's no biopsy until screening, but you don't really have screening until you have symptoms. So what we set out to do is we wanted to figure out what label-free biomarkers are available, because a lot of these samples, they're very precious. We don't want to waste them and section them. We want a high statistical power with as little biopsy as possible, to hopefully, the ultimate dream is to get like an optical biopsy going. But we're focusing on the morphological or the structural biomarkers and also the biomolecular ones. Oh, it's frozen. Let's see. Oops. Okay. And so what we do is we collect, as part of the HubMap program, many samples from, kind of control patients and diabetic patients, and kind of try to characterize them using, a few modalities that we see here. We can cluster them, we can identify them, and classify them. And in this section, I want to talk about the morphological features. And so we're very fortunate to have, glomeruli or glomeruli kind of in 3 distinct planes. So we have like a medial section because you can see that arteriole, but we also have very polar sections, and they'll all have a very different kind of ratio, like the glomerulus to the overall Bowman space. And this is kind of like an artifact of the traditional method where we do like lineal analysis. We have this section very thin on a grid, and you kind of count the number of pixels occupying the actual podocytes and the epithelial, like the tubules, and the empty Bowman space. But this can be done really quickly using modern image processing tools, like in ImageJ, or any segmentation model that you're using. But what you'll notice is that the ratio that people are using differ very significantly with each consecutive section. And so you have a very wide variation in any given 2D plane on your imaging slide. And so what we can do, since it's an optical platform, we can just scan in the Z direction, And luckily, depending on the lens you're using, a 200 micron glomerulus is, a perfect, like you can get at least one hemisphere of it to get a more accurate measurement. And so we found that a lot of the 2D, mesangial expansion analysis kind of underestimate um, the surface, the volume ratio. Uh, and so we are trying to, make sure that we can get as complete of a picture as we can in any given biopsy or necropsy, um, using as few glomeruli as possible. And so the other morphological analysis that we did in this paper is to look at the collagen fiber thickness. So basically, we transformed the second harmonic generation image into a 2D like a distance map, where each pixel is then replaced by its distance to the nearest background pixel. And you can see from the 2.5D images here, generally we see the same trends in the control versus the diabetic samples, where you have the cortex and the medulla very different, with the medullary ends having a very thick collagen, like a very dense collagen fiber matrix. But what we found was that when we look at specifically relative to the glomeruli, we can see that the fiber thicknesses in the diabetic sample, even though they're not like generally thicker overall compared to the control, they are more thick farther away from the glomeruli in the cortex, which was a new finding for us. This is very interesting. Still frozen. Let's see. Okay. And so a lot of pathologists might not be used to seeing these kinds of images, but since they're a hyperspectral image, like in a kind of like a 3D-ish image, we can just simply transform those images into a pseudohistology or a digital recreation of a histology. And I think some people have ascribed a name to it, like stimulated Raman histology. But so far, we're doing very well with typical H&E or PAS, which is great for typical like routine diagnoses. And we found that it also operates in 3D pretty well, so you can get 3D histology since again it's an optical sectioning technique. And it works pretty well with stock AI platforms. For example, we tried it out with the Halo AI by Indica Labs to identify, structures of interest. And you can see the top row is our Raman images that are recolored into the histology images, and the ground truth. They perform pretty well, but we also achieved very high classification accuracy and precision to identify the disease glomeruli. And again, so the optical redox ratio, bringing it kind of full circle to measure kind of the redox state of the tissue. We find that fibrosis is often an indicator for, organ failure, and there's a strong link between fibrosis and oxidative stress. And so at the very least, and again, so, with FLIM, you can get a better picture of these redox states. But at the very least, we can see, why they might, why this kind of modality might help in the classification accuracy of this disease. And so, I guess in summary for this paper, a lot of the stains and reagents that are used in today's, research landscape are, costly. They can also be toxic and require a lot of expertise. And when you're purchasing, so for example, when we started our lab, or when my PhD professor and I started her lab, I should say, it was during the pandemic, and sourcing from multiple vendors, it was very difficult during that time because of the supply chain was very sensitive, and a lot of the biomedical costs were prioritized, for like hospital settings rather than basic research. Additionally, there's a statistic that I found that where roughly 30% of histological staining fails. That could be for many reasons. I don't know if that's still true today. better men than me have done routine histology, but nevertheless, slicing is a laborious task and risks physical damage to the sample, and even adjacent tissue slices, just 10 microns apart, sometimes requires significant processing to kind of co-register them. But using our label-free platform, we can identify several biomarkers that have demonstrated high classification accuracy, including collagen fibrous thickness, lipid saturation, protein lipid ratios, optical redox ratios, and morphological things like mesangial volume fraction and collagen fiber thickness, just to name a few. Yeah, and so moving forward, we're thinking about applying this to other diseases and also working on the software side, developing new algorithms to identify, for example, lipid subtypes. And so it's what's great about stimulated Raman imaging or Raman in general is that the peak amplitudes of these chemical spectra are proportional to the number of scattering molecules in the sample. So it's a quantitative technique. And so what you can do is you can measure the reference lipid subtypes and then try to find where they are most similar in the sample directly, rather than kind of deconvolving those spectra into components and then trying to ascribe those components to biomolecules indirectly, because there's no guarantee that any, component spectrum is going to be like a physiologically meaningful one. And this is really great because since it's a label-free technique and non-destructive, you can do things like multiplex immunofluorescence to identify cell types of interest and then find those lipid subtypes within each cell type of interest. And so this is an example of a codex image that we did on the exact same stimulated Raman scattering image. And this becomes a really important tool where, the preservation of the spatial biology really matters. So, for example, senescence is where, a cell stops dividing. But those senescent cells, even though, they accumulate with age, they're still rare. So about 1% or less generally, you'll find at any given, time of your cells are going to be senescent. And so if you take adjacent sections, your senescent cells might not be in that same position. So it's really important that you look at this exact same sample using one modality as another modality. And that's really only possible if you have a label-free non-destructive technique. And so in this case, we applied it to senescent immune cells in the human lymph node, which was just accepted in Cell Press Blue. And I want to credit my colleagues at Yale for this work. Running short on time, but the other modality that we've conjugated with this platform is our dbit. And so dbit is our way to profile the entire transcriptome and epigenome simultaneously, very deep sequencing spatially. So what we do is we made these PDMS chips, and they have these microchannels down to 10 microns in width. We flow a bunch of oligo barcodes across the tissue, and then we repeat that in the opposite direction using another unique set of barcodes. And what happens is that once you have these barcodes A and B linked to the endogenous DNA and RNA, we can mix everything up and then sequence them and then map them back to where they were spatially, because it's a grid. And using the histology image and vision transformers, we can impute that to the entire tissue sample to get the entire very deep sequencing to every cell that we have, provided that we remain kind of at least largely within sample, or to get like a representative chunk within our dbit grid. And that's really important because the histology, since it's label free, can be done on the exact same sample, so you don't have that whole adjacent section struggle that you would have otherwise. And so rounding out the time, I'd like to acknowledge my PhD group in the Ling and Shi lab, as well as my colleagues at Yale